In the last video about movement control, we talked about how muscles work and how the organization of the motor neurons as they innervate these muscles can lead to rather, I'm going to call them unconscious movements. Like when you're taking a walk outside and you're, let's say, having a conversation with a friend or just kind of spacing out and thinking about something else that you're doing the next day, you don't really think about your walking per se, right? You just walk. And so there's a level of behavior control where you can really flexibly do a lot of different things with your body without paying much attention to how you're doing it. But of course, at the same time, we can use the same limbs and the same muscles and control them in entirely different ways by executing volitional movements and voluntary movements. So for example, even if running to get to the soccer ball doesn't take much thought, you're just running, you're thinking about the ball, you're not really thinking about how you're moving your legs. When you go kick the ball and kick it with a very pointed purpose, trying to get it into the goal, then you do have to think about, okay, so how do I move each and every one of my leg muscles so that I can execute this particular maneuver in such a way that I accomplish the goal I want. And so even though we're talking about muscle control, and it's the same muscles, and it's on the same limbs, and it's even mediated by the same by the same motor neurons. The concept of how do you initiate movements and how do you control movements in a planned and volitional way requires a bit more of a higher order control that goes beyond the level of the spinal cord, goes beyond the level of the decerebrated cat. So we talked mostly down here in the previous lecture about the local neuron circuits and motor neurons. We talked about how if you hook up a small circuit with a central pattern generator and a little bit of sensory feedback from a proprioceptor, you can actually get pretty robust walking. It is possible to get perfectly fine walking even with perturbations if all you had were neurons that are down here. We're going to be focusing on the next couple of layers up now in thinking about volitional movement and movement control. The first area we'll talk about is the cerebellum. So the cerebellum is super cool, and it's also this really beautiful geometric structure that is in the very back of your head. It's tucked below the cerebellum. It's often overlooked just because it's not on top. I think this is a bit of a snobbery on the part of a lot of neuroscientists because it's kind of on the bottom, and frankly, it's really hard to get to. And so sometimes we study things just because we can get to them, not because they're like more important than the deeper structures. This is the thing that happens. So the cerebellum is down there. And one of the reasons we know it's important is because we know it's required for fine control of movement. So when you have an animal with cerebellar defects, they exhibit ataxia, abnormal behaviors in coordinated movement. So what you're seeing here is the kind of tiny little footprints of a mouse. That's a normal mouse, a wild-type mouse. And you can see that its footprints are evenly spaced, right? And there's this very beautiful, perfect alternation pattern when it goes left, right, left, right, left, right. That's what's supposed to happen when we're walking. A mouse, instead, with a defective cerebellum, has the following symptom. It is no longer alternating between its left and right paws with an even spacing. You can see some of these left, left, left spacings are closer together and some of them are farther apart. It's unable to maintain going forward, even though I think it's intending to do so. It's kind of drifting to the side and then drifting back. You can also see by the imperfection of this little footprint. So I think the way they first do these experiments is just literally put, like, it's like fingerprinting, right? They put the paw of the mouse in some ink and then they had it walk on a piece of paper. And so you can kind of see, by the way, that these paw prints over here, right, are really smudgy and you don't see all of the little toesies on the little mouse. It's because it's holding its foot weird. It's holding it at an angle. And so it's unable to walk forward, it's unable to alternate correctly, and it's also unable to hold its foot and have it contact the ground in the way that it's really supposed to. And so all of those are symptoms of a cerebellar defect. So we know the cerebellum is required for fine control of movement. In terms of humans, you can have a similar symptom as well, where we see that individuals with cerebellar abnormalities are unable to execute smooth pointing movements. And so, for example, if I am instructed to lift my finger and then point at a spot in front of me, like a light lights up, I can point at it. So that's what happens when a normal individual, there's an acceleration, and then I slow down right before I touch the point, and that's what you're supposed to do. If you had a cerebellar defect, then the symptom is that these individuals might be attempting to do the same thing. First of all, there's the delay in the initiation of that pointing movement. And then if you track the trajectory of their movement, it appears to be uncoordinated. It doesn't go straight there. It in fact takes these meandering path and is kind of slow and unable to follow these smooth pursuit trajectories. So these are all pieces of evidence telling us that the cerebellum is required for fine control of movement. The cerebellum, like I told you, is the structure at the very bottom of the brain. It's kind of tucked underneath the cerebrum. If you ever have a chance to look at the anatomy of a cerebellum, it's absolutely beautiful. It's one of the brain structures that have the most regular structures. So it's just really pretty to look at. So that's what it looks like if you cut into it. Here is one of the major cell types in the cerebellum. It's called a Purkinje cell. This is a drawing by Ramon Hull of a Purkinje cell. I mean, back then, I think he cut it open and realized how beautiful it was. You can see this gigantic cell with this massive dendritic arborization, and it's kind of one-dimensional. There's these sheets of Purkinje cells, and the repetition of these sheets is what underlies the kind of fractally structure that is in the cerebellum. There's quite a bit known now about the cellular circuitry of the cerebellum. We have these Purkinje cells. This is a bunch of them all laid on top of each other. Their cell bodies are all in the same layer, lined up. Above them is what's called a molecular layer. Underneath is what's called a granule cell layers and other different types of cells. And so the circuitry that's repeated over and over and over in the cerebellum ad nauseum for filling the entire space of it is the following architecture. You have inputs, right? They come in through the cerebellum vestibular nuclei. And then there's these Purkinje cells. This is massive cell that's here, right? And they're innervated by these parallel fibers. And they also have what's called a climbing fiber that's climbing on top of here. It's like a snake climbing up a tree. That is a gigantic inhibitory set of synapses. And so this climbing fiber cell from the inferior olivary nucleus is able to shut down a Purkinje cell very, very reliably. And so there's a lot of theory about the learning that happens in the cerebellum that's mediated by this very fine circuitry that a lot of which you understand right now. Talking the details about it is beyond the scope of this particular lecture, but I did want to mention that this is a very active area of research, thinking about the computations in the cerebellum, as well as what are the implications of the cellular architecture and the cellular circuitry for the role that we know the cerebellum to play in fine movement control. So moving up, we have an area of the brain called the basal ganglia. And the basal ganglia, we also know, is roughly speaking, kind of waving my arms around a lot, important for the initiation and selection of motor programs. And so this is kind of a deeper part of the brain. It's not in cortex. It's kind of lower down. It's kind of the next layer of cells down. It's called basal ganglia. And it has a lot of different sub-areas that have different names. And so the point I'm trying to make here is that don't worry for now about the specific names of those specific brain areas. But they are there, and there's quite a bit of localization on modularity in the basal ganglia, not only in terms of anatomical segregation of different cell types and functions, but also in terms of cell types. So there's dopamine cells down there, there's serotonin cells down there, there's all kinds of local inhibitory neurons that are down there. The cellular architecture and the cell types in the basal ganglia are super interesting and kind of really complicated. Unlike the neocortex we talked about and the cerebellum that we talked about, different areas of the basal ganglia are not at all repeats of each other. It's not like you just made a bunch of copies and put them next to each other, like parts of neocortex look like that, and parts of the cerebellum most certainly look like that. That is not the case with the basal ganglia. It has a lot of different nuclei, and they seem to have distinct roles and do different things in different parts of motor program initiation and other different functions. Notably, there are two different diseases that are both disorders of the basal ganglia. So Parkinson's disease and Huntington's disease are both disorders of the basal ganglia. But looking at the differences between Parkinson's and Huntington's is really interesting because it tells us a little bit more about how the basal ganglia works. So here's another picture. Now, instead of looking at a human basal ganglia, we're now looking at a rat basal ganglia. And this is a sagittal slice of the basal ganglia, right? So anterior is over here, posterior is over here, dorsal is up, ventral is down. You can see the neocortex is up here, right? The neocortex is projecting down to the striatum and the basal ganglia, and the basal ganglia is projecting back up, these giant arrows of lots of pipes of information going up and down. In particular, there is two different pathways called the direct pathway and the indirect pathway. They are illustrated there by the two different shades of blue in this diagram. And what they do is that they actually invert the sign of the dopamine modulation. One of them is positively modulated by dopamine, and the other one is negatively modulated by dopamine. Now, exactly how this works, it's certainly beyond my ability to understand, to explain to you in one slide. But I do know that this is, once again, an area of extremely active research in terms of dissecting the fine circuitry of exactly how this works. Now, one thing I also put a pin in, just to make a connection for you, in case it didn't occur to you already by yourself, although it probably did. We're talking about dopamine here, and we talk about motor control and movement. The last time you heard about dopamine, it was in terms of pleasure and reward and the error signal. We mentioned this several different times. We mentioned it in the lecture about drugs and recreational drugs and drugs of abuse having to do with the dopamine system. Now, this is the same dopamine system. It's the VTA right there, the ventral tegmental area, and the substantia nigra, the SNC. Okay, same brain areas, same dopamine. Now, how does this work? I'm about to tell you about the role of dopamine dysfunction in motor control. And the last time you heard about the same dopamine, same brain areas, it was about an error prediction and addiction. How to reconcile these two very real roles that dopamine plays in your basal ganglia? Great question. I can't answer it right now. This is something that I might make a future video about frankly, after I educated myself a little more. It was always kind of confusing to me. I know that both of these things are true, and there's giant communities of people studying both. Exactly how they're reconciled, how they're the same thing, I don't quite understand right now. So I'm just, admitting my ignorance here. Yes, there's a connection. Yes, it's dopamine, but okay, fine. I do not understand exactly how they're connected and how they're working together. But in the context of motor control, we're going to talk a little bit more about the direct and the indirect pathways. And what we do know is that in Parkinson's disease, it is a disease, a neurodegenerative disease, where the person has difficulty initiating movements, and they have a resting tremor. So even when they're intending to not move, sometimes you'll have a resting tremor where they're unable to keep their limbs still. You can imagine this makes it quite difficult to execute everyday tasks, like picking up a spoon and feeding yourself would be difficult if you have a resting tremor. Individuals might also have trouble talking, for example. So we know that Parkinson's disease is associated with loss of dopamine neurons in the substantia nigra. And this imbalance, this loss of dopamine neurons, causes an imbalance in the direct pathway. And so because it's caused by loss of dopamine neurons, one of the therapies for compensating for the symptoms of Parkinson's disease is actually treating it with a dopamine precursor, L-DOPA. L-DOPA can be administered as a pharmaceutical. The body then metabolizes it into dopamine, thus compensating for the loss of dopamine in the substantia nigra. There's also ways of electrically stimulating this area of the brain by surgical electrical implants. We'll talk about that in 1 second. So on the other side, we have Huntington's disease, which is an imbalance that favors the direct pathway as opposed to favoring the indirect pathway. So it's also a motor movement initiation disorder, but it has a slightly different set of symptoms. We have instead uncontrolled movement of limbs, the head, and the torso, and it also associated with certain types of cognitive impairments. Huntington's disease is one of the more well-studied genetic diseases. As autosomal dominant, we know exactly which gene it lies on and how it is inherited. And so the loss of striatal neurons in the case of Huntington's disease preferentially favor the indirect pathway. And so to some extent, they have slightly overlapping symptoms. In Parkinson's and Huntington's are both motor disorders, where you have uncontrolled and undesirable movements of the body that the patient cannot control. But the way that they balance the indirect and direct pathways in the basal ganglia are opposite to each other. Unfortunately, as of right now, at least that I know of, there is no known treatment for Huntington's disease. So I'm going to talk a little bit about one of the really cool ways, cool in neuroscience sense, not cool for the people who suffer from Parkinson's disease, of treating the symptoms of Parkinson's disease. So I talked about L-DOPA as one of the treatments, and that actually works really well for certain people. Unfortunately, just like every other part of your nervous system adapts to new conditions. So if you have a loud sound, after a while you don't hear it anymore. If you have a noxious smell, after a while, fortunately, you don't smell it anymore. Similarly here, the body kind of maintains a homeostasis. And so after you get a treatment of L-DOPA, sometimes it causes only a temporary alleviation of the symptoms because your body then adjusts the new level of L-DOPA, and then that no longer alleviates the symptoms of Parkinson's anymore. So another really successful line of treatment for Parkinson's disease is what's called deep brain stimulation. This is a technology that I mentioned already in our lecture on electrical recordings and also electrical stimulation techniques in sensory and systems neuroscience. We'll just talk about it once more again here. So you have what's called the DBS electrode, the deep brain stimulating microelectrode. This microelectrode usually has something like 4 different contacts on it, and it has to go pretty deep into the brain. So it goes down like 10 centimeters or so because the area that's being targeted in the basal ganglia is quite a deep structure. A friend who is a neurosurgeon who was telling me that they actually spend an inordinate amount of time trying to figure out exactly how they target such a deep structure, because if you get unlucky, these basal ganglia structures are so small that if you deviate by a small number of degrees on the way down, the electrode will actually end up in a completely different place and will be ineffective in treating Parkinson's disease. And so they get really nerdy. They're nerd out about the small angle approximation when they're trying to go down the electrodes. And they're kind of floppy, they're really small, right? Because you don't want them to be too big. If you get too big, then they're going to damage structures along the way. So you kind of want to compensate for the fact that they're really small and thin and floppy, but then try to target them really precisely so you get to the right part of the brain for the treatment of Parkinson's disease. So anyway, DBS. Okay, so you have these DBS electrodes, and therapy goes the following. So the electrodes are going to be then passing pulses of electricity targeted at this area in the basal ganglia. To power this, what usually happens is that this is a Medtronic device made by the electronic metal biopharmaceutical company Medtronic, and they make this device where they're using the same technology that powers pacemakers for heart surgery and heart transplants. Where there's a chest implanted stimulator that holds the battery pack and also is able to program the exact series of pulses, like how frequently they come, how large they are, all of these parameters for the stimulation pulses. And they wire that up, and goes under your scalp, and then goes into the DBS electrodes. And so through this device, usually there's some kind of inductive charger, so you can kind of like sit there with a little charging pad, just like you charge your phone on a little charging pad. Same thing, you can do that with your chest implanted stimulator. Charge it, and then program it. This is something that is sometimes done by the user. They can kind of modulate their own implants if they want to turn it on and off, for example. And then the magnitude and frequency of the stimulation is something that one would usually discuss with one's clinician. And through trial and error, they would find out maybe, so like I said, usually there's 4 electrodes. And so sometimes they would say, oh, let's stimulate only through electrodes 1 and 2. And maybe for some other people, because they were off by a little bit during the implant surgery, they say, okay, let's do 2 and 3, right? So that's kind of a trial and error process in terms of which electrodes they're stimulating, how long, how often, and what the magnitude of the stimulation is. So here is a video of the almost miraculous effect of DBS in treatment of Parkinson's disease. Hopefully this video plays.
So we're here for the initial programming session.
And what we're seeing is, here's an individual with Parkinson's disease exhibiting these essential tremors. They're trying not to shake, but they can't help it, right? So they have this tremor of the arms.
Now we're just going to turn it on.
And you'll see that the clinician is now going to turn on the stimulation. Takes a second to press the button.
Fat fingers to hit the button. There we go. See how it feels.
And nearly instantaneously, the tremors stop. Not only do they stop, the patient is now able to speak and control their arms with an astonishing amount of precision that they clearly did not have before. The transformation is instantaneous and absolutely remarkable. So deep brain stimulation has been successfully used for treatment of Parkinson's disease, as well as it's slightly more experimental, but people have also experimented with DBS for treatment of mental illnesses, for example, including bipolar and major depression. So the mechanism of exactly why this works for treatment of Parkinson's disease is actually hard to explain. Like I talked about in the lecture about electrostimulations, we don't really know which cells are being stimulated when we pass current into the brain. It absolutely does work. This has now been done for enough individuals that it clearly does work in treatment of Parkinson's disease symptoms. We don't understand exactly why. So that remains to be understood. Okay, so we've talked about now the lower parts of this pathway, which I'm kind of labeling here as the execution pathway. So once you've decided what to do, this is the part of your nervous system that goes ahead and does it. So you've decided, I'm going to walk now. So you stop thinking about it, and it executes the plan of walking forward and not falling over. Okay, so that's mediated by your skeletal neurons, which are innervated by your motor neurons, which come from your spine, right, these spinal circuits. We've now gone up to the level of the basal ganglia and the cerebellum, which I think of as like the tactical centers of the brain in terms of movement. We're talking about initiating movements, figuring out when the movements are supposed to be, and then also inhibiting unnecessary and undesirable movements if that is what's appropriate for the circumstances. If you keep going up, we get to the area of strategy and planning. So the way that I think about this is using an analogy that I heard from a colleague, which is the kind of the mountain biking analogy. So if you're mountain biking down a trail, right, the bumps in the road are coming at you way too fast for you to actually thinking about it. Just like when you're hiking, if you're trail running, for example, if you're running on an uneven surface, the bumps are coming at you way too fast. You just have to execute and then deal with the bumps as they come. And that is something that happens at the level of proprioception and tactile sensation and spinal circuits. You just execute. But what's happening up here is a strategy and planning layer. So if you're mountain biking, you might see ahead of you that there's a giant rock, and it's going to be really horrible if you hit it. And so you're going to hit it in about 5 seconds, which means you have to plan for the future. You have to like veer slightly to the right and then maybe tilt your weight left or something so that you don't hit this rock that you can see. Now, that level of planning requires a much slower sensory modality, vision. Proprioception is super duper fast, okay? Vision is much slower. We're talking about milliseconds versus hundreds of milliseconds here. But the vision gives you information about the future in a way that's inaccessible from your somatosensory system. You just can't feel that rock that is 10 feet ahead of you. You can only see it. And so the advantage there is that you can see stuff that's ahead of you and plan ahead. The disadvantage is it's much slower, so you have to plan for how to deal with it before it comes. So that involves the cortical areas, not only motor cortex, okay, but also higher association areas. And this includes parts of premotor cortex and parietal cortex, a couple of other different areas that are doing this multisensory integration, integrating information from multiple modalities like vision and somatosensation, for example, and then also solving the problem of, among all the different ways I can avoid that rock that's coming up, which of them do I actually execute? And that depends on things like experience, like I know my bike and I know what it can do and I know what it can't do. I know my own strength, right? Like I know how much faster or slower I can pedal. I know how fast I can brake this bike. And so all of those are going to inform your planning of what you're doing at the next stage in your execution of these movements. So I talked to you in the last lecture about somatosensation, about the cortical organization of the somatosensory system. I told you that there's this giant chasm in the side of your head, one on each side, called the central sulcus. All of us have one. Right behind it is primary somatosensory area S1. Right in front of it is the primary motor area M1. Now this is the crossing over point where sensory information over here, so parietal areas are kind of higher order associational areas to do multisensory integration, and then go to the front. The more the front you go, these are more like the execution planning areas. Okay, M1, these are supplemental motor areas, prefrontal cortex, all of those are correlated with planning of movement, and then thinking about decision making, like deciding what to do, right? And then once these areas decide what to do. They then send that command down to the level of the thalamus, the basal ganglia, and the cerebellum, which then send commands all the way to the brainstem, which goes down your spine, and it ultimately ends up as a coordinated movement of your muscles. Now, there's an important point here, which is that it turns out in the motor cortex, this idea of a motor receptive field just makes absolutely no sense at all. People were thinking about it in this way for a long time, and there's a gigantic fight in the literature for decades because each individual neuron in M1, a motor cortex, they tried simulating it, they did a bunch of experiments, and they never quite found the map between one motor neuron and one muscle contraction movement on the contralateral side. The most parsimonious explanation for this is that it actually doesn't exist. We talked about population codes already in a couple of lectures ago. We talked about population code in terms of the three-dimensional representation of color vision, where you have to know the population activity of all three of those photoreceptors to figure out what the light is. We also talked about population codes in the olfactory cortex, because you have hundreds of olfactory receptors, and they all bind a little bit to a bunch of different odors. So the only way you can tell which one is it a rose or if it's a pile of stinky fish, is by looking at the population of activity of all of the odor receptors. Similar idea here, although it's kind of the opposite, right? The oppositeness is that it's the population activity of all of the neurons in motor cortex that drives the decision of what's going to happen in planning movements. And so by looking at each single one of these neurons, you don't gain that much information about what the plan is. You don't get a signature of what the decision is going to be. What is the kinematic trajectory that we're planning? That's not what it represents. So it's kind of like a receptive field, except for a motor neuron. And so instead of having a receptive field of a neuron, because again, that like intellectual picture just doesn't make sense in this context, this perspective instead says it's the population activity of these frontal and motor cortical area neurons that encodes these movement trajectories as dynamical systems. A dynamical systems, I mean that in the mathematical sense where you have dx dt as a function of x. And so it's this kinematic trajectories in the population activity that you can very cleanly and beautifully demonstrate that it's decoding of that dynamical system that gives us a clue of what the animal is planning, because we can decode from that activity and figure out what it's going to do next. Okay, so another application of the idea that that's just one of the reasons you have so many cells in your brain is because it's not each single one actually matters, all right? There's no one motor neuron that matches to one muscle. That's just not how it works. You don't think about which muscles do you move. You think about where you're going. You think about, okay, I want to reach a coffee mug over here, and I'm gonna go for it, right? I'm gonna plan my trajectory of reaching for the coffee mug. At no point did I actually think about all the different muscles in my arm and my back that need to move for me to execute this movement. That's delegated to the execution stage of the motor pathway. I'm just thinking about the path. I'm thinking about I'm going to go to the coffee cup, and I can decide to go this way, or I can decide to go this way. So I do decide that, right? That's something to which I have conscious access, but I don't have access, nor do I need to have access to the execution phase of that movement. Okay, so I'm going to summarize here by talking about some of the themes that I've hopefully have brought up over and over, not only in this lecture about motor control and motor initiation and planning of movement, but you've seen a lot of these themes in a lot of different lectures in the section of sensory motor control. I started out by telling you about these action perception loops and how this type of feedback is absolutely crucial to understanding the functions of the nervous system and behavior. And so what I've shown you in terms of the neural circuitry of not only the sensory but also the motor systems is that feedback is absolutely central, not only between the animal and the external environment, but within the nervous system at every scale of description. We've seen feedback loops from proprioceptive neurons onto motor neurons. We've seen feedback at the level of local circuits in the spine. And there's feedback at the level of basically every single scale of description all the way up, right? This is similarly true for sensory systems. The only place where there's not sensory feedback is between the retina and the thalamus. The thalamus doesn't project back to the retina, but that's it. Like everywhere else, there's a ton of feedback. There's feedback between different sensory systems. There's feedback between different motor systems and sensory systems so that we can see signatures of movement in even what was previously thought of as sensory areas. So feedback is a really crucial feature of all of systems neuroscience, and one in which we cannot ignore if we wanted to understand how it works as a whole. The second feature that we cannot ignore is this idea of hierarchy. We talked in the summary of the sensory neurobiology lecture about these canonical pathways of sensory information going up from the primary sensors to the thalamus to the cortex, and then back in the motor system down to the thalamus, the brainstem to the spine, going back and down, right? So all of these systems are hierarchical, and they differ by timescales. The lower down ones tend to be obsessed with executing things. They have super low latency, really fast. You often don't have access to them consciously. You don't think about it, you just do it. And then there's these higher cognitive areas where you do things that are slower, that require more deliberation, but are kind of commanding the lower down areas. But a lot of it is delegated. You don't think about exactly how you're moving the muscles in your arms. You think about that you want to do it and what you want to accomplish instead. And so that is the feedback and the hierarchy. And these are just two themes that we see a lot in systems neuroscience at every scale of description in both sensory and motor systems. The last theme I'll emphasize here is that we talked a lot in separate lectures about the different sensory systems. But I hope you already noticed in this lecture in particular about motor control, like I told you about the mountain biking example, that for real behavior, for you to actually interact with the world in the way that you normally do, requires multi-sensory integration. So very quickly, after all of this information gets into your central nervous system, they all get mixed up, and all of the different sensory streams are modulating each other, and they are combining with each other to allow us to form a full sensory perception of the world on which we're going to make decisions about what we're going to do next. And so this is happening in a hierarchical way, and with feedback. And so perhaps on theme, these three themes are not really separate either, because the way in which we execute multisensory integration is through hierarchical systems that feedback on each other. Okay, so those are the themes. I hope that this has been a fun journey through the sensory motor systems. There's a lot more to talk about here. But the next time you're walking down the stairs, I strongly encourage you to not think about which muscles are moving in what order. I tried doing that one time. Literally the first time I learned about muscles and how muscles actually work in muscle control, I tried thinking about what my legs were doing as I walked down the stairs, and I almost fell. So don't do it. Don't think about it. Just do it.